And you got to admit, this is like the coolest title you ever saw, right? It's got bird poop in it. So this will be a paper by Jennifer Deaver and Jody Rowley talking about uncovering species within the bird poop frog complex in Southeast Asia.
Thank you so much for hanging around. Hopefully you got a chance to get a little bit of coffee during that little break. That's great. I'm Jennifer Deaver, and I'm from the University of San Francisco. I'm also a research associate at the California Academy of Sciences. And my collaborator, Jody Rowley, is at the Australian Museum. She's got loads of expertise in the area of Southeast Asian frogs. I have been doing some work on the Amelops complex in the past, and now we've been collaborating for the last little over a year on this project, uncovering species within the bird poop frog complex. So today I'm going to basically tell you a little bit what the motivation of our research was, why we did it. I'm going to talk to you about how we discovered the diversity within this complex, and I'm going to tell you about the conservation implications of our findings. So first of all, why are we interested in Southeast Asian frogs? And I think it really comes to the fact that Southeast Asia is a really cool place, but more importantly, we're all interested in the global amphibian crisis. It's a global phenomenon, yet there are regions that do warrant more consideration in the world, more so than others. And specifically, this idea of biodiversity hotspots came about in 2000 by Myers. These are regions of high endemism that also have high levels of habitat degradation. There are 4 such regions within this area. And our research is focusing now primarily on the Indo-Burma and Sundaland regions. So Sodhi in 2004 actually wrote about the diversity of this area. And basically he talks about how the complex geologic history, the interesting topographic features, the heterogeneous vegetation, and the tropical climate all combine together to provide for a substrate for great diversification and a lot of species. In just the Indo-Burma region, there are actually 32 different ecoregions that have been identified. And within these regions, 271 described frogs that are endemic to this region exist as of today. But this number is actually relatively low compared to other parts of the world. For example, if you were to count up the number of frogs, endemic frogs in Central America and Colombia, which is a square kilometer region that's about the same size as this region, there are over 640 endemic species. So a lot more species have been described in those areas. Why is that the case? Researchers such as Brown and Stewart in 2001 sort of talked about how there's been not enough descriptions going on in this area, that basically the diversity has been significantly underestimated. And Raleigh in her PNAS paper actually talks about the reasons being, there's a lot of logistical issues with actually sampling in this area. There's also been a history of political unrest, and the expeditions that have happened have been basically sporadic and very localized. And so the fact that there's a lot of undescribed species that are present here, and the fact that this is an area of sort of interesting concern for us from an evolutionary standpoint, has really motivated us to take a look at this area. But what's really sort of lit a fire under our butts is the fact that it is an area of serious habitat destruction. So in that same paper I was talking about, Sodhi actually talks about this full-fledged disaster, because over 75% of the forest is actually projected to be completely destroyed by the year 2100 if nothing happens. And that will actually lead to potentially nearly half of all the biodiversity going extinct. So this is a real crisis. And again, it was described as sort of this perfect storm, because you've got illegal logging, you've got basically unchecked economic development, corruption, the drug trade. All of these things are happening at a very fast pace in an area with a lot of biodiversity. So if something isn't done soon, we're about to lose a lot of these undescribed species. So this is the motivation behind that. But then why Theoloderma? And when I talk to Theoloderma, everybody thinks about the mossy frog, Theoloderma corticale. But we're interested in the bird poop frog, because who doesn't love the bird poop frog? I mean, come on, it's great, right? This is a tree frog in the family of Rhacophoridae. Since it was described in 1886 by Boulanger, it's not a lot to know about it. I mean, it is kind of interesting. It's widely distributed, found throughout Southeast Asia, and basically it's really hard to find. and there's not much that's known about it. Although it's somewhat common in the pet trade, we have hardly any real information about its natural history. So that begs us to think, oh, let's look at this in a little bit more detail. So when we thought about this, we started to look at the different sort of phenotypes that are out there. And these are, again, known as members of Philoderma, basically the fact that they don't have webbing between their fingers, they're relatively small in size, and they have some degree of tuberculation on the skin, and they kind of have this, patterning that looks like bird poop. But there's a lot of variation within the patterning. And again, based on Brown and Stewart's postulation that frogs in Southeast Asia that are widely distributed probably are made up of many species and not just the single species, we hypothesized that these are probably members of a cryptid complex. And so to do that, we went out, we looked at frogs that were in collections, so we examined specimens and we did some DNA analysis. So first of all, the recent exhibitions really got us going. California Academy of Sciences did a long, multi-year study of Myanmar, where several specimens were collected. Jody actually has engaged in multiple expeditions to Vietnam, but I should note that only 9 frogs were actually collected from these multitude of expeditions. Again, speaking to the rarity and the difficulty of finding these frogs. We also were able to get our hands on 18 different Philoderma asperum specimens from collections around the world, including the Sin type from the British Museum. And we also were able to look at additional Philoderma species for comparison. So we took 12 morphological measurements for each specimen to the nearest 10th millimeter. We also examined each of the individuals to see if they had nuptial pads or vocal sacs, and we looked at the skin texture as well as the coloration for each. For the DNA taxonomy, we sequenced the mitochondrial DNA, the 16S region, of about 487 base pairs of that, and then 4 additional nuclear genes were sequenced for a total of 17 plus 100 base pairs for a subset of 18 individuals. So we were able to then align these sequences, compare them to the closest relatives in GenBank, do the usual phylogenetic analysis that you've been hearing so much about today and this weekend. And then our results showed something interesting. First of all, from a morphological standpoint, we were able to see subtle differences in the coloration. So if you take a look here, we have 2 frogs from different regions. And just note, I just want you to note there's this eye patch here. The shape is more Y-shaped here. Here it's a little bit more of a small triangle. It's really subtle. There's also more of a sort of a D-shaped curve here. This is a little bit more pointed here. And there's some orangish coloration on the side, which is not on the side of this guy. So there's slight, very subtle differences in the coloration. But when it came down to the morphological measurements, there were no significant differences whatsoever. And another kind of interesting thing, 2 of the male frogs, one from Chin and one from Kachin in Myanmar, did have vocal sacs, whereas none of the other frogs had vocal sacs, which is an interesting difference. The DNA shows us a little bit better picture. And here we have the results from the Bayesian phylogeny. The maximum likelihood tree typology is exactly the same, so we're just showing Bayesian here. And if you'll note, you have all of the Philoderma here. They're all forming a monophyletic clade. And Nyctixilus is actually falling out here as basically the closest sister taxa to this group. And then here I just want to show you, of course, these are all the Philoderma asperum specimens. So there is a lot of diversity that is exhibited here. All of the internal nodes have high support. How does this line up with the nuclear subset? And you can see that there is congruence between the 2 trees. There is some slight differences, but that's not to be unexpected because they do evolve at a different rate. But the pattern is very similar. So if we zoom in to just those asperum specimens, what we can see here is we have 8 distinct lineages with pretty decent divergence here. And again, interestingly, we have Philoderma lysine, which is actually within this sort of unresolved group of asperum. So that was a somewhat interesting finding here. So when we look at this, we wonder, okay, what's the haplotype network look like? And a pattern kind of emerges whereby those that have the shared haplotypes are also geographically closest to one another. So we see this patterning that's occurring, and that led us to kind of take a look back at that ecoregion map and see, hey, how do these fall out? And it turns out, for the most part, by and large, the lineages actually fall within distinct ecoregions. And so, as you can see, for example, here's the Indo-Malayan forest that are clustering here. Here we have these guys that are all clustering along the Annamites rainforest and so on and so forth. There are some exceptions, just to point out this guy. This is a specimen from the Yunnan, and it is falling in a different ecoregion than these, but yet they're kind of grouping up together. So here's that guy there, and then here are the rest of these. And then also, these are our Kachin frogs, and you can see one's in this region and another's in this region. But notice the geographic distance. So they are kind of far apart, and if I kind of direct your attention, again, here they are in the clade, and there is a bit of distance genetically between the two. So what about the genetic distances? We have values that are above the 3% threshold that Miguel Vences suggests for the 16S, ranging from 4% to 16%, so very high levels of divergence. And then again, if we take a look and see, can we see any phenotypic variation that lines up with these clades, we start to see some patterns. So there is some visible difference, but it's very subtle, doesn't just jump right out at you. So the question here is, can we identify these frogs without the DNA? That's always the big question. And I will say that here you can see they do kind of group together. You can see, for example, the Laos frogs have this kind of distinct, more triangular patch. And again, the coloration is very similar with kind of a lighter color at the top. We see more of a pointed pattern here. The northern Vietnamese ones all have more of a Y-shaped patch and a broader curve here with, again, more gold coloration to the sides. And then for the central Vietnam, we see some very distinct, this kind of orangish patch is common in all those. Another thing that I found really interesting is that looking at a paper from Li and Hu that was written in 1962 in Chinese, they described Philoderma albopunctatus. Now, you may have not heard of that, but it was a legitimate species up until 2008 when Yu sunk it and synonymized it. And he based that synonymy basically on just 3 samples of the 12S, 16S gene, one from Guangxi, one from northern Vietnam, and one from Yunnan. And they did group together with the other Philoderma, one asperum that he had. And so he said, you know what, it's Philoderma asperum. However, translating the Chinese was no easy task. I had to ask multiple people, but finally, they have vocal sacs. The Philoderma asperum do not have vocal sacs. Another interesting thing we see here is that the pattern, this is from the illustration that's provided, it does look a little bit more like those northern frogs from northern Vietnam, which are closest. And again, the genetic clade, they're grouping all together. But an interesting point is these males do not have vocal sacs. So, in conclusion, we have shown that we have a cryptic complex here, and that there's at least 6 known species that we're going to end up describing, potentially more, but we do need additional samples before we can do that. We're going to add some CT data to see if we can actually see any cranial characteristics, and it would be great if we could actually look at those Chinese specimens. But the real thing here I want to leave you with is that this shows how there's a lot of hidden diversity in this area. And again, what's going on in Southeast Asia is not an enigmatic phenomenon. It's not like, what's happening? I don't know. We know what's happening, and we need to put all our effort into getting good scientific data to describe the species that are there so that we can help manage what we have before it's too late. So I hope I've motivated you guys to all switch over to Southeast Asian frogs. And if I have, then I've done my job. I want to go ahead and thank all of the great folks at the various museums who have been so gracious to send me tissues and samples. Thanks for all of our funding. And of course, we can't forget Lauren Scheinberg and good old Jens at the California Academy of Sciences. So with that, I'll take any questions.